All right, so welcome everyone, and thank you so much for taking the time to come to our first ever online workshop for HNN. And thank you all for filling out that pre-workshop survey. That's really useful for us to improve our workshop and to report back to the NIH, who is funding this workshop, that things are going well and there's room for improvement, and we will move in that direction. So, and I also want to thank Medicel for hosting us. They're providing all of the computational resources that we're going to be using during the workshop, and they're also organizing our event this morning online. So thank you, Paulo and Zoran. So let me begin by introducing you to the HNN development team. HNN is developed by many institutions and now by many people worldwide. We're an open source software, and we're trying really hard to follow best practices in open source software design. And so we now have lots of community developers from all around the world. But the main HNN development team was a team out of Brown University, the Martino Center at MGH, and the Neuron team at Yale. And I'm pointing out some specific people here that are here today to help run the workshop and really have been core developers of the HNN software. And so that's me at the top. I'm a professor in the neuroscience department at Brown University, and my lab has created this software, the initial parts of the software, and now, of course, it's grown in many directions. I have two graduate students here, Nick and Ryan, who will be helping with the workshop and running some of the hands-on tutorials. They're both graduate students and lead developers of the HNN software. Dylan is our project manager, and he's also a software developer. And then Minok at MGH created HNN Core and has been really spearheading. HNN Core is the Python version, the underlying code to all of the HNN software, and Minok's going to help run the workshop as well. And we have a special guest, Kaisu, from MGH, who is going to present on a recent study that she published using HNN and kind of give you a case study walkthrough of her experience from beginning to publication. So thank you to the team. We're funded by the NIH, and thanks again to Medicel. So all of these people are here today, and you can ask any one of them questions as we move forward. So the schedule, we have a very packed schedule for the next 3 hours, but hopefully that means you'll leave here knowing a lot more than you did before we started the HNN workshop. So I'm going to start by giving you an overview of HNN, about what it is, the background information on how to use it, a little precursor to the hands-on workshops that we're going to be walking through, and then we'll have a little bit of time for question and answer. Then we're going to go for about an hour into breakout rooms, and we're going to do hands-on studies of event-related potentials and how you get started thinking about using HNN to study the mechanisms of generation of ERPs. And we're going to take a short break. And then Kaisu is going to come and give a short talk on her experience with HNN and some details from the recent publication that they did on multisensory integration, looking at integration of auditory and visual evoked responses. And then we're going to have another shorter hands-on workshop where we go over how we use HNN to start to study rhythmic activity, alpha rhythms and beta rhythms. And again, we'll have 3 breakout rooms there. Then we'll have a brief introduction of HNN Core, which is the Python interface. Again, HNN Core is the underlying code for everything. We have a graphical user interface, so you don't ever have to look at the underlying code, but there's more features and advanced functionality in HNN Core, and Nick will talk a little bit about that. And at the end, we're going to try to leave 15 minutes and ask you to please take another survey about the workshop experience. Again, this is really important for us to be able to report back to the NIH, who is funding the dissemination and continued development of the software. So we really appreciate your feedback. You can be anonymous, you can be good, bad, whatever it is, please just be honest. Okay, so I'm going to get started. If you have questions as we go along, please just put them in the chat, and I can either be interrupted if it's a really major question that's causing confusion, or we can just answer all the questions at the end. So an overview. So I'm going to begin by giving you the background on the development of HNN, what it is and how we went about developing. Then I'm going to go through 2 case studies that we used in the lab, which forms the basis of our tutorials. The study of sensory evoked potentials, and in our lab, we look at somatosensory perception, and the study of alpha and beta rhythms, again, in the somatosensory cortex is where we've been studying. And then I'll briefly describe some other HNN applications and how to get started if you're not in the workshop, right? Today, we're going to run them on MetaCell's resources, but all of the software is downloadable. All of the tutorials are online, so we'll just review that very briefly. Okay, so background. So we developed HNN to address a big challenge in human neuroscience. And this challenge is that EEG and MEG are hugely powerful technologies because they give us direct readouts of human information processing with millisecond resolution. But a downside is we still lack understanding of how these so-called macro scale signals are generated by the cells and circuits within the brain. And this circuit level understanding is really critical if we want to know why these signals correlate with information processing, or how to target treatments when they're disrupted in diseases with either pharmacology of non-invasive brain stimulation. And so we created a computational neural modeling technique to bridge this gap. And of course, invasive recordings in mice or monkeys or any animal model and the types of neuromodulation that you can do in rodents is really powerful to dissect this cell and circuit level activity, but that's still really difficult in the human. And so having computational modeling that can bridge that gap is a really powerful way to start to make predictions about the detailed circuit origin of these signals. So there are two modeling techniques in order to get from the sensor level signal outside of the head to a cell and circuit level interpretation inside the head. The first modeling technique is called current source estimation modeling, where you take the sensor level signal and you estimate the location and the time course of these big electrical currents in the brain that are generating these sensor level signals. Once we know where and what those currents look like, we can interpret the cell and circuit origin with computational neuromodeling, where we simulate the electrical activity of the individual neurons in the circuit. And with this level of detailed modeling, we can have interpretation both at this microcircuit cell level, but also at this macro scale recording level. And so this is the foundation of HNN. HNN is not doing the current source estimation. We rely on other softwares like M&E or Brainstorm, whatever your favorite inverse solution software is. And then once you have an estimate of that electrical current, that's when you use HNN to interpret how was that electrical current generated. And for over a decade now, we've been turning this into a software, and now we have funding from NIH to distribute it to the community and to support the community in its use and development. So how do we go about creating a model that can connect these sensor level extracranial signals to the underlying cell and circuit level generation? Well, as I mentioned, the first step is to apply an inverse solution method. And this estimates the location and the direction and the time course of these big electrical currents. We refer to them as primary currents. These are what are creating the recorded signal. And so then we need to think about, well, where are these primary currents coming from in the neural circuit? Well, the primary currents are generated by the postsynaptic intracellular current flow and these long and spatially aligned cortical pyramidal neuron dendrites. Essentially, these dendrites are aligned in such a way that they create a massive electrical current that can be recorded outside of the head with either EEG or MEG. The generator of EEG and MEG is the same. It's the intracellular current in the pyramidal neuron dendrites. What's different is what reflection of that current you're measuring. You're either measuring the electric or the magnetic field, but they're coming from the same primary source generators. So given that this is where the signal comes from, there are some key canonical features of cortical circuitry that we wanted to take into account in order to study the cell and circuit level generation of this signal. The first is that the cortex is a layered structure. We have these pyramidal neurons with these long apical dendrites in both the supragranular and the infragranular layers. And they're synaptically coupled to other excitatory inhibitory cells within the local cortical circuit. These networks don't sit in isolation. They're constantly receiving inputs from other parts of the brain. And there's two primary pathways of information flow into cortical circuits. The first comes from the lemniscal thalamus. This relays sensory information from your body in the periphery into the thalamus and then up to the granular layers of the cortex where it propagates to the other layers. In the model, we refer to this as proximal drive because it effectively hits the proximal dendrites of these large pyramidal neurons. The other pathway of information comes into supragranular layers. This is either from higher order cortical circuits, or there's also non-specific thalamic nuclei that project right up and broadly to the supragranular layers. In the model, we refer to that as distal input because it impacts the distal dendrites of these pyramidal neurons. And then in the model, the primary current signal comes from just summing up this big intracellular electrical current in the pyramidal neuron dendrites. And with this construction, the units that come out of the model are current times distance, nanoamp meters. That's the same unit that you get from your source localized EEG or MEG signal. So we have one-to-one comparison between the output of the model and our source localized data. And this proves to be a very powerful way to start to develop and test predictions on how these signals are generated by the underlying circuits. So we know a little bit about the size of the networks that contribute to these most commonly measured EEG and MEG signals. So what are we commonly measuring? We're measuring an ERP, or we're measuring a low frequency oscillation, alpha, beta, theta, gamma. ERPs typically tend to be about 10 to 100 nanoamp meters. They're thought to be generated by the orders of tens of thousands of synchronously firing pyramidal neurons. Low frequency rhythms are even bigger. They're about 100 to 1,000 nanoamp meters, and they're thought to be generated by on the order of 1 million pyramidal neurons in its sub-threshold current flow across these pyramidal neurons. Not that 1 million pyramidal neurons are firing action potentials at a low frequency rate. So just to give you a little bit of detail, I'm not going to show you any equations here. All of the equations are available in the underlying code that's distributed online. But the way that we create this network is we have multiple compartment pyramidal neurons, and we have these single compartment inhibitory neurons. In neuromodeling, we call them point neurons. And the reason we collapse all of the structure is because these neurons aren't contributing directly to the recorded signal, but they're nonetheless hugely important for the dynamics of what goes on in this underlying network. We have GABAergic and glutamatergic synapses between the excitatory and inhibitory cells, GABA A, GABA B, AMPA, NMDA, and we simulate the electrical activity in each and every compartment of these neurons using a Hodgkin-Huxley current balance equations. We have a capacitive current equal to a sum of ionic currents. We have sodium currents, potassium currents, calcium currents, all sorts of active currents in our neurons, and they were tuned to reproduce realistic behaviors that we know about in these cells. And then this primary current dipole we get by using a cable theory equation. And again, we just sum up this intracellular current flow between the compartments. Now, in practice, when you're using HNN, we're not simulating all of these other brain areas that are interacting with HNN. We have a local patch of cortex. And what we do is we simulate trains of action potentials that activate excitatory synapses in the local network and these projection patterns. And that creates activity and drives current flow up and down these pyramidal neuron dendrites. So for example, if we have a feed-forward drive, or a drive that comes in from the limnoscal thalamus, these action potentials, we call it either proximal or feed-forward, they're going to activate these synapses and they can drive current flow up the dendrites and create an upward deflection in your current dipole signal. On the other hand, we can simulate a train of action potentials that hit the network in a feedback or distal projection pattern. This will drive current flow down the dendrites and create a downward deflecting signal. And it's the interaction of these feed-forward and feedback drives with the local network dynamics that create this big current that goes up and down the pyramidal neuron dendrites. HNN was specifically designed to simulate activity from a localized area. It represents a little patch of cortex. And we've been developing it around workflows to study these common signals, ERPs and brain rhythms. This is all based on our prior published work. And we give you our data and we give you our starting parameters, but it's meant to be a hypothesis development and testing tool. So if you have another idea in your data about where these signals come from, then you can test that using this software. And again, we're just building it around a starting point for people to be able to develop and test hypotheses. In order to make this is a large scale neural model, right? There's thousands of differential equations. There's even more parameters. In order to make it accessible to the broad MEG and EEG community with money from the NIH, and in particular the Brain Initiative, we embedded all the underlying code in this graphical user interface. And so to get started, you don't need to look at the code at all. We have a website that contains installation instructions and tutorials on how to simulate ERPs, alpha, beta, and gamma. Again, we give you the starting point, we give you a tuned model, and we give you an idea of how we did it, with the idea that then if you learn that, you can go and do it on your own. We also have a Python interface, which is called HNN Core. And the tutorials that we've developed for HNN GUI, we're also developing for HNN Core. So the idea is once you know how to do it in the GUI, if you know something about Python coding, then you can go in and do the same thing in HNN Core. And there's more functionality in HNN Core, which we'll talk about later this morning. Okay, so sensory evoked potentials. As I've mentioned, in my lab, we study sensory processing. And the first study that we did back in 2007 is we said, okay, we're going to give a strong tap to the finger, and we're going to look at what was the mechanism of generation of the ERP that comes from that tap. And then we're going to give a threshold level tap. Sometimes you feel it, sometimes you don't, and we're going to study correlates of perception. And that's what we did in this 2007 paper. So what I'm showing you here is the evoked response from this brief tap to the finger when the subject feels it all of the time, super threshold, and when they feel it half of the time, threshold. And this is source localized to the hand area of S1. And so we used HNN to ask the question, what is the circuit origin of these early components of the tactile evoked response? And how do the circuit dynamics change between this super threshold and this threshold level response? And we were able to reproduce this in our HNN model and develop a lot of predictions on what was going on in the underlying circuit. So that's what I'm going to walk you through. And in the hands-on tutorial, we're going to go through this super threshold evoked response generation. Okay, so the first key thing is if we want to relate this current signal to the underlying cell and circuit level activity, we have to have a sense of what is a downward versus what is an upward current, right? In our signal. Is a downward current going down the dendrites, or does that reflect current flow up the dendrites? And so you want to know which time points of the waveform current flow to current down and up, or in and out of the cortex is another way to say this. And so to do that, you use your inverse methods. And you can apply an orientation constrained inverse method to determine the orientation at the peak, right? And here's an example where we used M&E with a fixed orientation. If you use a whole label, then you want to flip the sign of the current so that it's all going in the same direction. So now we know that this peak is corresponding to current flow down the dendrites. That's a really important thing when you're trying to connect it to cell and circuit level activity through HNN. Is this up or down the dendrites? I'm going to show you that for sensory evoked potentials, we have a good idea that the first peak is always up. And so if you're doing sensory evoked activity and you know this is the first peak of your signal, you have a pretty good understanding that that's going to be a current flow up the dendrites. Okay, so how do we constrain this model to study the neural origin of the sensory evoked response? Again, this is a really large scale neural modeling. Well, there is a method to the madness. The first thing we did was we built the individual's neurons. We fixed all of the anatomy and all of the physiology based on a lot of animal work studying the anatomy and physiology of cells across the cortical layers. This has been built mainly on information from the primary somatosensory cortex, but we found that this cortical canonical circuit has been can be applied to study other brain areas as well, because these signals outside of the head are reflecting this macro scale structure that's conserved across brain areas. Then we built in the local connectivity. How are these cells connected across the cortical layers? Again, there's a lot of information on this. The only thing that we started to do when playing around with how this signal was generated is we started to vary the timing and the strength of these exogenous inputs that we have control over that activate the local network. And by playing around with the timing and the strength of these inputs, we were able to develop and test hypotheses on where these signals come from. So let me walk you through how we did this for the somatosensory evoked potential. The first thing we did is we load in our data, right? And you can do file, load data, and here's our very robust S1 evoked ERP. Then we went to the literature. And there's a lot of literature on how are sensory evoked potentials, ERPs, generated. And what the literature suggests that there's a sequence of activation to the local cortical circuit. At approximately 25 milliseconds after you tap the finger, you get a feed-forward input, a barrage of input to the local cortical circuit that comes through this feed-forward pathway. About 70 milliseconds after you tap the finger, there's a feedback input. That signal has gone somewhere else in the brain and then comes back into the local cortical circuit through these feedback connections. Then there's a thalamocortical loop that's been generated, and it comes back up in this feed-forward prediction pattern at about 125 milliseconds. And again, this hypothesis is based on a lot of animal work trying to understand the origin of these ERPs. And so in the model, we can define a sequence of inputs that follows this projection pattern and see what does that do when I activate this local network. And so what we do in the software is we have predefined parameters. And so you can do set parameters. We're going to go to the evoked input parameters, and we're going to distribute the sequence of input that we gave to the HNN model to simulate this ERP. And you load that in, and now you see it. So this is a histogram of spikes. There's a barrage of spikes that come in. And this is the timing and the distribution of those spikes. Here's the feed forward, here's the feedback, and here is the late feed forward. Now, we spent a lot of time tuning the strength and the distribution of these inputs in order to test the hypothesis that these could generate this source localized ERP. So then we run simulation, and it can very nicely reproduce this. Again, the literature gave us a sense of where to begin. And then we spent a lot of time tuning the parameters of the model to see if we could get an accurate representation of the signal. And once you do, now you have all these underlying predictions about the details of how this was generated. And you can go into the model and you can view the layer-specific dipoles. You can view the spiking activity of all the neurons. You can view the power spectral density. And all of these features let you, one, test these model predictions with invasive recordings in animals, but they also give you a sense of how does the microcircuit activity create this macro scale signal that I recorded outside of the head. And so it's an interplay between looking at the macro scale signal and the micro scale dynamics, where you start to get a sense of how these peaks are generated. And that's what we're going to walk through in our hands-on today. In HNN Core, we also have the ability to simulate local field potentials across the cortical layers and the current source density. And again, all of this is really important in order to validate the predictions that are coming out of the model, and then to adjust the model if our predictions aren't supported by invasive recordings. This tool is supposed to be a tool that bridges between macro and micro, between human work and hopefully animal work and validation of our predictions. Okay, so let me just briefly walk you through what we're going to, again, walk through in the hands-on tutorial, but how is this up and downward current flow generated by this circuit and that sequence of input to the local circuit? Well, the first thing we can look at is, well, what's going on in the different layers? We see in layer 2, 3, which has these pyramidal dendrites, you get just this small current dipole. In layer 5, you get a bigger current, and then the aggregate is just the sum. It's just the sum of what's going on in layer 2, 3, and 5. It's just what's the whole network doing. And you can see that layer 5 contributes more. And that's because layer 5 has longer dendrites. This is current over distance. And so you tend to get a bigger signal from layer 5 than you do from layer 2, 3. Now, if layer 5 is not active and only layer 2, 3 is, then layer 2, 3 is going to dominate your signal. But in general, layer 5 creates bigger responses. We can look at the underlying spiking activity. So what's going on in the basket, which are the inhibitory cells, and the pyramidal neurons, which are the excitatory cells, and the supragranular and infragranular layers. And you see that there's a lot of underlying structure here that's hard to understand when you just look at the macro scale signal. But what we've learned through simulation is that outward currents or upward current flow, right, up the dendrite, can come from either proximal depolarization pushing you up the currents. If you get action potentials in these cells, they backpropagate up the dendrites. If you have inhibition out at these distal dendrites, that will pull current flow up. This figure is coming from a recent paper by Ryan Thorpe, who is one of our HNN developers. And I encourage you to grab this paper because this is really essential when you start to think about, well, what's causing current flow up and down and how does it relate to spiking activity in the layer-specific responses? Inward currents or downward current flow can come from either distal depolarization, so you get an excitatory current that can push current flow down. It can come from, we have calcium in the dendrites. If you get a dendritic calcium spike, that pushes current flow down. Or it can come from inhibition at the soma. If you have inhibition at the soma, inhibition will pull you down. If you have inhibition at the distal dendrites, that will pull you up. And so in general, these are the things we start to think about in controlling the upward and downward current flow in our ERP. So what's going on here? Well, at the beginning, we've got this pyramidal neuron spiking that's pushing current flow up the dendrites. Then we have inhibition in a lack of spiking. Well, that's somatic inhibition, so that pulls current flow down the dendrites. Then we get some reemergent spiking activity, which pushes current flow up the dendrites. Then this distal input comes in, pushes current flow down the dendrites, along with calcium activity. Then we get a bunch of spiking, and that's all that is back propagating up the dendrites to create that positive deflecting signal. And so by playing around with looking at the macro scale and the micro scale, you start to get a sense of how is this circuit creating this macro scale signal. Now, in practice, we have some optimization tools. So we're going to give you a pre-tuned model, right? We're going to give you preset values, and then you can go in and play with those values. And in practice, usually what you do is hand tuning is invaluable because it just gives you a sense, well, what if I change this? What happens? What if I change that? Once you have a close representation to the signal, you can use our automated optimization algorithms. And so if you go into simulation configure optimization, you can choose the parameters you want to optimize, the range to optimize around. You run optimization. Here's the fit before I optimized. Here's the black is the fit after one optimization. I can optimize again. Here's another optimization. And then ultimately, I get to the fully optimized solution. Now, this is going to give you one solution, right? It's going to give you a value of all the parameters that can produce this. But there may be multiple parameters that equally fit this signal. And we're working right now, Nick Tolley, who's one of our developers, is developing methods to find distributions of parameters that might account for your signal. And we have a recent publication on that, and that will be something that becomes available. Okay. The other thing we've shown is that evoked responses in other sensory areas, so this is our somatosensory, this is our auditory, this is our visual, that same sequence of information flow, feed forward, feedback, feed forward, can account for the ERP in all these areas. And so what that suggests is that the macro scale signal is really dictated by commonalities in the cortical structure in these brain areas that we're capturing with HNN. And they represent this sequence of activation that pushes current flow up and down. And so some of the take-home messages from these studies is that ERPs are generated by a sequence, or can be generated by a sequence of proximal, distal, and proximal drive. The first peak, as I mentioned, is current flow up the dendrites. This largest peak is current flow down the dendrites. And so again, if you have a sensory evoked response, you can be fairly certain that that first peak is up the dendrites. Okay, how do the circuit dynamics change between superthreshold and threshold level stimuli? Well, here, what we did is we tested a hypothesis. We said, okay, if I tap the finger very strong, then I get this ERP, and I've fit my model to this signal. Well, what happens when I give a threshold level stimulus? Well, maybe what happens is the inputs come in and they're just weaker, right? They're not as strong coming into the local cortical circuit. And so we tune down the conductance strength of the inputs. Oh, sorry, I meant to mention the scaling factor is really important. So this network has 100 pyramidal neurons in the supragranular and infragranular layers. That's 200 pyramidal neurons. But the size of this signal is on the order of 100 nanoampere meters. In order to get that size signal, we had to scale the output of this network by 3,000. So we took the nanoamp value out of the 200 cells, we multiplied it by 3,000, and now we're on the same scale as our data. And so there's a prediction there that on the order of 60,000 pyramidal neurons or 600,000 pyramidal neurons are contributing to this macro scale signal. And the assumption is synchrony, right? That a lot of the population is doing a very similar thing. Okay, so now we turn down the strength, and by just, this is the same pattern of drive, but by just weakening these connections, we were able to reproduce the features of the tactile evoked response. And importantly, there's some timing differences here that emerge naturally out of the network dynamic when we just turn down the strength of those inputs. And again, we can go in now and look at the spiking activity and see what's different in the underlying network. Okay, I'm going to briefly go through our study of alpha and beta rhythms. So before we tap the finger, in the somatosensory cortex, you get this robust low frequency oscillation as components in the alpha band and in the beta band, 10 hertz and roughly 20 hertz. On any single trial, they're very bursty or event-like. It's only when you average across many pre-stimulus epochs that you get these continuous bands of activity. So in HNN, we wanted to say, what is the circuit origin of these transient alpha beta components of the S1U rhythm, right? Because on any 1 second time interval, we get either an alpha or a beta. So how do we get that stochastic nature of this rhythm? And it's just showing you that with HNN, we can reproduce many of these features, and on average, we do get these bands of activity. So how did we do this? Well, again, we go back to the theory. There's a lot of study on the origin of these rhythms in animal models and in prior modeling work. And prior theories on the mechanisms of generation of alpha is that there's an alpha generator in the thalamus, and it comes up and it hits the cortex does something, interacts with the thalamus, and so you get these thalamocortical loops of activity that generate alpha. And so in the model, we can generate 10 hertz input from the thalamus and see, does that produce alpha in our model? Beta rhythms were thought to be generated by these specific currents in pyramidal neurons that when interacted with excitation and inhibition, they create these 20 hertz oscillation. If anybody out there knows about ping, pyramidal interneuron gamma, this is just a slowed ping due to these M currents. And so we can build these M currents into our model and ask the question, if I drive it at alpha and I engage these M currents, do I get this alpha beta complex? And what we found was no, that wasn't the case. So if we just come in and we hit this network with a burst of input every 100 milliseconds coming from the thalamus, what that does is it drives current flow up the dendrites, and then it comes back down the dendrites, and we get a beautiful alpha oscillation. But this doesn't look anything like our actual signal, which goes up and down and presumably reflects current flow up and down the pyramidal neuron dendrites. And so that said to us, well, what if we add another input to the distal dendrites? That should start to push current flow down the dendrites and break this upward current flow. And through trial and error and lots of simulations, we found that if we had two 10 hertz thalamic inputs that come into the network in these proximal and distal projection patterns, and on average, they come in simultaneously, right? The timing of these could be separated, they could be together, but if on average they come in simultaneously, we get this alpha and beta rhythm. So this was a brand new prediction about how beta can be generated in cortical circuits. The alpha part kind of fits with the prior literature, but the beta component was brand new. Again, we have a scaling factor here. One of the important things about our alpha beta simulation, this is sub-threshold. I am not driving these cells to spike. I'm assuming that these alpha and beta rhythms are coming from the sub-threshold activity of a really large population of neurons. For the ERP, I'm multiplied by 3,000 to get the right amplitude. Here, I'm multiplying by 30,000. Lots more neurons in a sub-threshold regime contributing to this response. And we quantified all sorts of features in our data that we were able to reproduce with this model simulation. Now, again, this was a new prediction about where this S1U rhythms comes from. But because of the level of detail in the model, we could test a lot of these predictions. And we now have validation about the model from predictions from laminar recordings in V1. This isn't a paper by our group, but a paper published by Charlie Schroeder's group. And then we have laminar recordings in mice and monkeys. and also high resolution MEG, which are starting to validate. Again, this is a hypothesis development testing tool. You come up with a prediction, you want to validate it. Laminar recordings are really essential for validation because we're integrating information across the cortical layers. And so ultimately to test these, we want information across the cortical layers. Okay, very briefly, what are some other applications? We also have tutorials on gamma rhythms. We're not going to be going through this today. Gamma is a very robust signal that can be recorded with MEG and EEG. I'm just showing you one example here, and that we can reproduce this with HNN. There's a tutorial online. I encourage you to look at it and do it if you want to learn more about the generation of gamma rhythms. There's lots of published examples of using HNN to study various signals. We've studied on brain rhythms we've published on brain rhythms on the alpha, beta, and gamma bands. We've also looked at sensory perception and tactile perception and auditory perception. We have some motor evoked potential papers as well. We've looked at biomarkers of pain perception. This recent article from Ryan is about laser evoked compared to median nerve stimulation. Other groups are looking at pharmacology for depression. We published on non-invasive brain stimulation, changes in ERPs and rhythms with aging. Other groups have looked at changes in children with autism spectrum disorder. And we have a few studies where we've started to combine human imaging, HNN, and animal work to try to validate some of the predictions that are coming out of the model. You can see the full publication list on our website. So how do you get started? Well, you can install it on any operating system, the GUI version of this code. You can also run it through the Neuroscience Gateway portal. The Neuroscience Gateway portal is a supercomputing center at the San Diego Supercomputing Center, and they're providing resources for the neuroscience community to run computational neuroscience software. The GUI isn't working there yet, but the HNN core version is. The GUI should be working very soon. We're creating a new graphical interface at this moment. There's a lot of challenges with sustaining the installation of the current GUI, and those challenges are hopefully going to go away with our new GUI, and so that will be on the portal. HNN Core is our Python interface. You can install it with one line of code, pip install. Again, we're going to have a new GUI in HNN Core soon, developed on HNN Core code, but both Core and, HNN Core can be run both on your local machine as well as right now on the San Diego supercomputers. Tutorials. We have tutorials on alpha, beta, and gamma based on our prior studies, also ERPs. We have similar tutorials in HNN Core Python. Again, the idea is you learn it in the GUI, and then you can reproduce the same steps in Python code. And we have some example how-tos on how to get started with the Python code. And again, there's more functionality in the Python code than there is in the graphical user interface, because we just can't build everything into the graphical user interface. We have a Twitter, please follow us. Okay, and so now I just want to I know we're a few minutes over, but let me just take a moment to answer any questions that have come up, and then we're going to go into breakout rooms and launch the software. And this is where we're going to go to launch it with your email address and that password. So any questions? I know that was kind of a whirlwind intro to HNN, but as we go through hands-on, it'll become clearer and clearer, I think, to everybody. You'll have more opportunity to ask lots of questions. Do we have any hands up? I'm not seeing all the Zoom people.
I have a question.
Yes.
This is Maruti. I tried to put the hands up, but somehow it was not working in mine. Maybe I didn't know. So my question is,
No
I see that we can do the rhythms in using HNN. So is this rhythm part very specific to ERPs, or even resting state can be done?
You can test hypotheses about either. What we're distributing, what we're going to walk through today, is about resting, spontaneous pre-stimulus rhythms that we've studied in the lab. But you could study evoked rhythms. You just have to have an idea about where those evoked rhythms coming from. Is it the same mechanism of generation of the spontaneous, or do I think it might be a different mechanism of generation? But what we're distributing is based on spontaneous, our prior published work. Yep.
And I have another question. What about the inferior brain areas, like the folded parts? No. Okay.
Oh, that, yeah, you get cortex, right? We're estimating signals coming from cortex, not subcortical brain structures. But the folding and where you are in cortex, that all you determine through your inverse solution methods. And so once you have an area of the brain and you know where it is along that cortical folding and what the signal is from that area from your inverse methods, now HNN is a tool to interpret how that signal was generated. It's not doing the inverse of where is the signal in the brain.
Cool. Okay. Thank you.
Thank you. Yes, again, I'm having maybe if I stop sharing, I can see everybody. Sorry. Does somebody want to just shout out?
I had a quick question.
Yes.
The scaling factor, would you expect any difference in the outputs from the models if you actually ran a huge number of neurons versus applying a scaling factor from the output of a small
Absolutely.
number?
Yeah,
Okay.
each neuron is contributing some nanoamp current. And so the more neurons you have, the bigger potential current you can get. So you would expect to have a smaller scaling factor, absolutely.
Well, I mean, is there a difference between scaled output from a small number of neurons versus unscaled output from a very large number of neurons? Like interactions between the neurons, would that affect what you're seeing with an actual, actual full-scale model?
Yeah. So you're getting to a good point, which is the heterogeneity across a larger population. And how much of that do we get by just scaling? We don't just scale, and this is something we'll go through in the hands-on workshop. We also do a smoothing. So to account for, we've got only 200 neurons in our network, and there's some kind of stochasticity in who's firing when. But in the brain, there's lots more neurons and a lot more stochasticity to get to this thing that we record outside the head that's really pretty smooth, right? And so to get that smoothing and account for that stochasticity, we apply a smoothing factor, assuming there's jitter across a larger population, and that jitter averages out when you record from everybody. So I can't answer your question exactly, but my guess is when you have a bigger population, the smoothing will come from averaging the jitter across that population, and you'll have less of a smoothing factor in the HNN simulation. But it's a really good question. I don't think we know the exact answer to that.
Cool.
Yep.
Maybe it would be fun to run a full-scale model on the supercomputer.
Absolutely. Yep. Xenia?
Yeah, thank you very much. I have two questions. So first of all, do you also have tools to do quantitative model fitting? From what I understand, you mainly look at curves and compare the shape, but is there also a tool to do some correlations between time resulting ERPs or anything like that?
No, but you can download all of the output from the model and do any kind of correlation studies on your own. And again, in HN Core, it's open source. So if you develop a new feature, share it. That would be fabulous.
And the second question is it possible to do source estimation without an available structural MRI scan, but just from the standard locations of the EEG electrodes?
Again, HNN doesn't do source localization, but if you really want to understand where the source is and what the directionality of the current is, you will want an MRI. But I say that we've applied HNN to sensor level signals because we have an understanding that if it's a strong sensory response, and I record that outside of the head, I know that first input's a feed forward input. And so I can start to guess if I can assume that I'm looking at an early response that hasn't spread and I'm not, EEG has all sorts of field spread issues. If I'm pretty sure that it's, coming from a localized circuit and I get a robust response, you can still start to use HNN with sensor level signals. The challenge is what is up and what is down out when you record outside of the head.
Thank you.
Yep.